Okay, so when the question was asked, I'll get you. So, question was asked, where do I find the exam review sheet? And if you scroll here down the modules to the week 4 learning activities module, scroll down to the bottom, study Dr. Evers exam 1 review sheet. Okay, so if you click that, so you can see the whole document, but it has in here what you know for chapter 1, chapter 2, chapter 3. Also tells you here these possible short answer essays, because remember on the exam, be mostly multiple choice, but you'll have some short answer where you got to type an answer, some some where you got to match, right? And you'll have one essay question. And so this is one potential essay question. Okay, here's another potential essay question. And then another potential essay question for chapter 3. Okay, so, and it tells you, generally speaking, what's going to be on the exam. Okay, so that way you know exactly what you need to study. All right. So we're getting ready to start chapter 3 today. So talking a lot about cells and permeability, osmosis, solutions, organelles in the cell, parts of the cell cycle, mitosis, meiosis, all right, DNA, RNA, transcription and translation, protein synthesis, the genetic code, historical persons and what they're known for, concepts of cell theory, differences between prokaryotes and eukaryotes, okay, somatic and sex cells, definition, characteristics, and functions of parts of the cell membrane, types of transport, okay, diffusion, parts of the cell cycle, how many cells you get at the end of mitosis, factors that affect diffusion rates, okay, explaining transcription and translation. All right, if you're given the nitrogen bases to a sense strand, which is your template strand, and we haven't covered this yet, but we'll cover it in this chapter. So write the corresponding nitrogen bases of mRNA. And bonus, if you're given the nitrogen bases to a sense strand of DNA, can you write the corresponding nitrogen bases in mRNA, the coding strand or nonsense strand of DNA, as well as the tRNA, which is the anticodon. Again, none of that you may know today, but it's stuff that we'll go over. So I actually just decided to put the review sheet right at the top of week 4, so that way you'll easily be able to find it. All right, so with that, let's start chapter 3, cellular level of organization. So in order to think about that, we've done the chemical level, right? So we started the lower level, now we're moving up to cellular level. Okay, so let's start with what is a cell, right? They said, what was his name? I think it was Vince Lombardi. It started out with his football team to make sure they were really understanding the fundamentals of the game. And he said, okay, this is a football, right? So we're going to start with this as a cell. So it is the structural and functional unit of living organisms, okay? So while we have smaller levels, we consider it to be life at the cellular level, okay? Before that, we consider it kind of just components, right? So some important people that you need to know about in their contributions. These are things that tend to show up as matching, right? Match the person with their contribution. So Robert Hooke in 1665, he described chambers in cork tissue and called them cells. This is the first time anybody used that term, the cell. All right, so that's That's a big deal in science, okay? 1838, Schleiden proposed the idea that all plants consist of cells. And you might say, well, that's pretty basic, but in 1838, people didn't know that, okay? In 1839, Schwann put forth the idea that all animals consist of cells, all right? So Schwann said, okay, Schleiden, I'll do you one better, right? So, okay, plants are cells, animals are cells too, okay? So know those three, okay? Know what they did. All right, contemporary cell theory. Okay, I think this was one of those points that was in that review that you needed to know the components, okay? So cells are the building blocks of all plants and animals. Well, that comes from Schleiden and Schwann, okay? Cells are produced by the division of pre-existing cells. So when cells divide, that produces new cells. Cells are the smallest unit that perform all vital physiological functions, right? So remember, it's our smallest unit of living organisms. Well, I shouldn't say of living organisms. It's the one where we say it's life, okay? We'll say it that way. All right, and each cell maintains homeostasis at the cellular level, okay? Homeostasis is at the cellular level. Homeostasis, or balance, equilibrium, at the tissue, organ system, and organism levels. It's going to reflect combined and coordinated actions of many cells. All right, so what's happening at the cellular level determines what's happening at the levels above it. Does that make sense? Okay, so if you have a disease, you see it at the organ system level or the organism level, but there's something also going on at the cellular level, okay? So that's kind of what the point that they're trying to make. So, a question like this might be, write down 2 of the 5 components of cell theory, okay? So the more you know, the more likely you're going to get that question right? All right, studying cells. So studying cells involves cytology, which is the study of the structure and function of cells. Remember, ology is study of, cyto means cell. It's part of cell biology. Now, there's 2 different kinds of cells, right? We have prokaryotic cells and eukaryotic cells. Okay, I think this was also one of those, characteristics. You want to be able to answer a question regarding these characteristics of prokaryotic and eukaryotic cells. So you might have to list 2 or 3 characteristics of both, or let's say you list 2 ways that prokaryotic cells are different from eukaryotic cells. And so, you might say prokaryotic cells lack a nuclear membrane in one box. The other box for eukaryotic, you'd say has a nuclear membrane, okay? For prokaryotic cells or bacterial cells, you'd say they lack most organelles, which are the kind of cell parts. Eukaryotic cells are going to have those organelles, okay, most of them. And they're going to have, and when we say eukaryotic, we're talking about the cells in your body. We're talking about animal cells, plant cells, protocells, okay? Smaller in size for the prokaryotic cells, that the eukaryotic cells are larger in size. So again, like how this might look is you've got boxes that you've got to type an answer in, and you'd literally be typing in what it says here, okay? But I also want to make sure you remember you're going to have a box for both, right? So you're going to have a box for prokaryotic and a box for eukaryotic. So you got to be able to explain it for both. And on any writing question, you might get the question right, and because Canvas is not a human being, it might count a writing question wrong, because it says, like, I have to type in answers. If you type in it's a letter different or it's said in a different way, it counts it wrong. I have to go back in after the fact and change it to a correct answer, okay? So when you take the exam and it gives you a grade at the end, know that the portion of it that is short answer or essay hasn't been graded yet. And so some of those short answers might get converted to a right answer, and you haven't got any credit on your essay. Again, on the essays, do the essay. In the Start Here module, I have a rubric that shows you exactly how to get 20 out of 20 or however many points that essay is worth on that test, how to get 100 on the essay. I'm not asking for a article, a research article. I'm asking you essentially for maybe a few sentences, okay? You give me the correct information and it's grammatically correct, you're good, okay? So it is very, very possible to get full credit on the essay, and you don't have to be a genius to get it, okay? You just follow the rules, okay? So that is what I want you to do. So just, and we'll show you that next week, but I want you to be aware that there is, an essay. It's not, when I say essay, it's not essay for English class, okay? Essay for me is like 4 or 5 sentences, okay? So, because as they used to say on the internet, ain't nobody got time for that, right? So, just want you to be able to write clearly and concisely, okay? All right. So for prokaryotic cells, they have one singular circular strand of DNA. It's just in a circle, okay? For the eukaryotic cells, you're going to have many strands of DNA, and they're going to be in the form of chromatin, which basically is DNA that's wrapped around protein. That's really all that is. All right, and that forms your chromosomes. Last difference, prokaryotes are going to have small ribosomes, and they are different in structure from what you see in the eukaryotes. Eukaryotes, because you have all this compartmentalization, you find them in eukaryotes on the endoplasmic reticulum, and we'll talk about that more later. Or they can just be free floating in the cytoplasm, okay? So those are important differences between eukaryotic cells and prokaryotic cells. All right, so, all right, what are the 2 types of cells? Prokaryotic and eukaryotic. Which one's bacteria? Prokaryotic. Which ones are like human body cells? Okay. Which one has a single circular strand of DNA? Okay. Which one has DNA that's wrapped in chromatin? Okay. Which one lacks a nuclear membrane? Which one has most organelles? Okay, that's what I need y'all to be able to do, okay? All right, so cellular anatomy. Human body has 2 types of cells. It has sex cells and germ cells, and it also has somatic cells, okay? So most of your body cells are considered somatic, which means essentially they're not sex cells, and they help your body form, okay? Pretty much any cell is not a germ cell is a somatic cell. And germ cell or sex cell is a fancy way of saying eggs and sperm, okay? So our sex cells or germ cells are reproductive cells, okay? So involved in sexual reproduction. And they exist at different stages, okay? So when we say egg and sperm, those are generally we're talking about more mature sex cells, but you kind of have some more, they're immature primordial cells or immature gametes, but usually when we're talking about mature gametes, that's a term that we use for eggs and sperm. But again, somatic cells are your body cells, sex cells, or germ cells are your sex cells, okay? So which are your normal body cells? And which are your sex cells? Okay, other word for sex cells is germ cells, okay? All right, so body's got a lot of cells, 100 trillion. Okay, you don't see them all because they're so small, but you got over 100 trillion cells. So, you know, like they say on Austin Powers, like 1 million. You know, so you have 100 trillion cells, right? So all of our activities result from the combined and coordinated responses of these millions and billions and trillions of cells. Okay. That being said, even though you have so many, you've got around 200 different types of cells. So they all fit into one of these 200 categories. But most of these cells have the same parts in them. Okay, they just serve different functions. All right, and gives you here table 3-1. Okay, the page number is kind of irrelevant because you have an e-text, most of you. All right, but you're going to look for table 3-1, but we're going to cover the different types of or parts of the cell. All right, so I just want you to be aware of that piece. So the page numbers are from the hardcover. All right, cell membrane. We'll start there. So our outside of the cell is going to be all this extracellular fluid. In this case, when we say extra, we really mean outside. Okay, so it's kind of a watery substance, surrounds the cells. On the tissue level, we call it interstitial fluid. Okay. Cell membrane or your plasma membrane serves as your boundary for your cell. Okay, it keeps certain things in, certain things out. So what are some functions of the plasma membrane? Again, this is another good short answer. You know, give me 2 or 3 characteristics of the cell membrane type of thing. Or which of, or multiple choice, which of these is or is not a function of the cell membrane, right? So always think about it when you're studying, like, well, how might this show up as a question? You know, and you kind of come up with that. So functions of the cell membrane. And again, cell membrane and plasma membrane are interchangeable, okay? Physical isolation, right? It's a physical barrier. So it's separating your inside of your cell from the outside of the cell and the fluid outside of the cell. All right, regulation of exchange with the environment. So what does that mean? So it's just controlling whether or not certain ions, certain nutrients can get in or out of the cell. It helps you eliminate waste from your cell. So just like you eliminate waste from your whole body, your cells eliminate waste too, okay? You're also releasing secretions out of cells, okay? So your cell membrane is selective or sensitive. So it reacts, it's one of the first to react to changes in your extracellular fluid. So it's going to react before the inside of your cell. So if you have a change in pH, remember that's like acidity or basicity, changes in temperature, your cell membrane is going to react. And it does that because it has receptors, right? So, you know, think about it like if you're, you know, on a, you know, fort or you're on a, you know, ship, or you know, if you're in the military or something like that, like you've got, you know, people that are stationed to let you know, hey, so and so is trying to get close to, you know, wherever we're trying to do, and then they send that information back to, you know, their folks on the inside. That's essentially what happens on your cells too. All right, so like, oh, hey, you know, we see this infectious disease that's hanging out outside of your cell. You know, you know, so there's going to be a reaction that happens on the inside to protect your cell, right? So structural support. So it's going to give stability to your cells and your tissues, right? So if you didn't have a cell membrane, all this stuff would have, it would just, you know, go everywhere. It has to have a container to keep everything where it's supposed to be. All right. So those are important functions of the plasma membrane. All right. So let's talk a little more about that membrane, its structure. So it's around 6 to 10 nanometers thick, so pretty thin, right? What do you have in it? Well, some of those good organic molecules that we studied in chapter 2. So remember lipids? Hopefully you remember lipids. Remember proteins, right? Carbohydrates. Like, oh yeah, we talked a little bit about that last week. Okay. So, and if like I'll say it like I say in the video, if you don't remember, I've got a video about that. You can watch it. So, phospholipids, they are the biggest contributors to the structure of the membrane. Okay. Your membrane is what we call a phospholipid bilayer. So, what does that mean? Well, what is, bi means two, right? Like a bicycle has two wheels. It's a double layer of phospholipids, right? So phospholipid molecules, and we study this in chapter 2. Remember, they have a polar head or a hydrophilic head. It likes water, and it's on the surface of the membrane on either side. So it touches the outside of the cell, and then there's another head on the other layer that touches the inside. All right, those hydrophobic tails, those non-polar tails, are going to lie on the inside of your membrane. Okay, so what that means is water and solute molecules can't cross the lipid portion. Okay, so the inside of that membrane doesn't like water, so water can't get through. Right, so you have to have some way of being able to get things through, which we'll talk about in a minute. But that's an important piece to separate your cytoplasm on the inside of your cell from what's going on outside your cell. And so just to kind of show you, let's just say that my hand is one phospholipid. This is part of one layer. So like the base of my hand is the polar head. My fingers are the two lipid tails, phospholipid. And then you've got another layer, okay? And they're just, sitting on each other like that. And it goes all the way around in a circle, okay, or a sphere. So that essentially is kind of how your phospholipid bilayer works. All right. So in the membrane, you've got some proteins, all right? And they could be on one side of the membrane or they could go all the way across, okay? So you've got what we call integral proteins, which are integral or important parts of the structure of the membrane. You also have what we call peripheral proteins. And these are attached to the membrane. They're kind of like on one side, but they're not really a piece of the membrane, right? They just sort of get tacked onto it, right? So membrane proteins serve some roles. And again, these are good kind of either short answer or multiple choice type questions, but they can serve as anchors, right? So they can attach your cell membrane to some other structure, okay? So, an anchor, you throw out the anchor, docks your boat, right? So it's similar concept. You can dock that cell connected to something and stabilize the position. All right, the membrane proteins, okay, they get bound to the cytoskeleton. Again, I said, what did I say cyto means? Anybody remember cyto? Cell. Yes, very good. Skeleton is your skeleton. So it's like an internal skeleton inside your cell. Okay, provides support. All right, their membrane proteins can be identifiers or recognition proteins. Okay, so that's how your immune system recognizes other cells as either being normal cells or abnormal. Okay, so you have to be able to differentiate between a normal cell and the cell that's infected in your body, or a cell from a microorganism that's not one of your cells, or a cell that is cancerous or something like that. Your body has to have a way of differentiating those cells, and those receptors on the outside allow you to do that. Okay. Enzymes. Remember enzymes in last chapter with proteins? Remember they catalyze chemical reactions, right? So you have receptors, okay? And so these are sensitive to what we call, and it should say ligands. They're sensitive to ligands. And they will bind to them and change the activity of the cell, okay? So it's similar to what we talked about with the recognition proteins, seeing if something is bad, but you have receptors that really are looking out for specific things that you might need, okay? So similar in that respect. All right. Carrier proteins. So as the name suggests, they basically either form a highway and allow, or a bridge, allow things to come across, or they pick them up and move them to the other side, okay? So the carrier proteins are more of the pick them up and move them to the other side, okay? The channels are more of the expressway or the bridge, okay? So, and with the channels, it allows things like water, small solutes to get across the membrane, okay? Because again, water is not going to be able to go across on its own. So channels can be described as either leaky or gated, okay? And if they're leaky, means basically you can get through all the time, okay? And I would liken it to kind of a highway where you just get on the highway and you drive. I would liken the gated channels to your streets where they got street lights, okay? Sometimes red light, sometimes the green light. Okay, if it's a red light, they can't go through, okay? Same kind of concept. All right. Membrane phospholipids in proteins. So, well, the membrane and its phospholipids in proteins is considered to be fluid. All right, that's an important word you want to know, fluid. What does fluid means? It's not solid, okay? Molecules can move around, particularly laterally, which means they can move along the membrane, okay? Proteins are not uniformly distributed, okay? So it's not a rigid pattern, which means the proteins are in a mosaic, okay? So that word mosaic is important for you to know. And I'll make this a little larger, but mosaic is important because what if you ever seen a mosaic, it's just a bunch of different things mixed together in no particular order, right? So composition of that cell membrane changes over time because of what we call metabolic turnover. And I know, we throw a lot of terms at you, but you got to break them down. So when we say metabolic or metabolism, we're just talking about chemical reactions, okay? So proteins or materials in your cell or in your cell membrane, they typically don't last forever, okay? They use them up for a while, then they get destroyed, then you got to make new ones, okay? You get rid of the old stuff and put in something new, okay? That's basically what that is. All right, inner and outer surfaces of your cell membrane can have differences in the protein composition and the lipid composition, okay? So just what it looks like, what your membrane looks like on the outer surface doesn't have to be the same as what it looks like on the inner surface. They can be different. Same basic components, but they can be organized differently. So we call this the fluid mosaic model, okay, of your plasma membrane. Right, so it's important to know that term, fluid mosaic. All right. So at least now you know where those words come from, fluid mosaic model. All right. So carbohydrates are in the membrane too, not just proteins. All right. So the glycocalyx is one of those carbohydrates. And it serves these functions. It protects and lubricates your cell membrane, serves as an anchor for your cells, helps in motion for certain cells, locomotion. You might be moving those cells from somewhere to another, or it could be just moving some part of the cell, okay? Glycocalyx can also function as a receptor, and part of that glycocalyx is recognized by your immune system. Okay, that's part of that normal versus abnormal. Okay, it's involved in that. And characteristics of the glycocalyx are genetically determined. Okay, so there's some genetic role in terms of regulation here. And that comes from your DNA. So at least that, while this is carbohydrate, it does have some impact on what's happening with the glycocalyx. All right, so we tell you all this to tell you about permeability. And so I think what I want to do is pause for a minute, and I want you and your battle buddy to talk about characteristics of the membrane, as well as membrane proteins and membrane carbohydrates. You can do this at home as well. So now that we've covered parts of the membrane, let's go ahead and talk about permeability in the membrane. So what is permeability? Well, it's basically how easily substances can get across the cell membrane. Now, membranes could be impermeable, which means nothing can get across. They could be freely permeable and anything can get across the membrane. Or they can be selectively permeable, where some materials can get through and others cannot. And our cell membranes are selectively permeable, okay? So definitely want to know the term selectively permeable, right? Now, movement across that membrane can be active or it can be passive. If it's passive, no energy is required to get those molecules across. If it requires energy, typically in the form of ATP, to move the molecules across the cell membrane, then it's active. Now, there are some categories for membrane transport. We have diffusion, we have filtration, we have carrier-mediated transport, and vesicular transport, okay? Diffusion, filtration, carrier-mediated transport, and vesicular transport. All right, so what's diffusion? Well, it's moving material from an area of high concentration to an area of low concentration. Okay. All right. So in order to understand that, you need to know what a concentration gradient is. So a gradient is basically a difference. Okay. So it's the difference between that high concentration and the low concentration. So if it's a big difference, there's a big gradient. Small difference, small gradient. Okay. So what's diffusion in that context? Well, that means that diffusion is going to happen until you eliminate the concentration gradient. So the goal is to get to equilibrium, okay? Now, when that happens, it doesn't mean there's no movement. It just means the movement is equal in both directions, okay? So diffusion is going to typically eliminate your local concentration gradients in your body, okay? So that's the goal. All right, so I think one of your, well, I shouldn't say I think, I know one of the parts of your, or questions on that review was factors that influence diffusion rates. So this could show up as, what are three things that, or three factors that influence diffusion rates? So again, the more you remember, the more likely you are to get it right. Okay, so distance. So if you have a shorter distance to get across, diffusion is going to be faster, right? So I mean, you think about if you had to run, you know, if I say, okay, run across from here to there, or run a mile, which one are you going to do faster? From here to there, right? Because it's a shorter distance. Size of the gradient. You have a bigger concentration gradient, you have faster diffusion is going to proceed because you've got it. Just, I mean, basically, it's kind of an urgency. When you have a bigger concentration gradient, you've got more pressure. Okay, so you're going to have faster diffusion when you have a bigger gradient. Okay, so molecule size. So ions and small organic molecules are going to diffuse more rapidly than larger proteins. So again, if I've got to run a mile, am I going to be able to run that mile faster with or without a rucksack on my back? Without, right? So you're putting on more weight, it's going to make you move more slowly. Okay, so temperature. So if you increase the temperature, you're increasing energy. Okay, make those molecules move more. So you're going to make diffusion happen more quickly. So electrical forces. So you have negative charges on the inside of your cell membrane are going to pull positive charges from the extracellular fluid, right? So if there's a difference in charge, you're going to actually be pulling the opposite charge, okay? Which can make diffusion happen more quickly. And that leads us to what we call an electrochemical gradient, which is basically a net or a total of your chemical forces and your electrical forces that act on an ion, because both of these things are happening at the same time. So if you have an ion and there's a certain amount of the ion on one side of the cell membrane and a certain amount on the other side, there's a concentration difference, but there's also a charge difference, okay? And both of those things kind of have to get to equilibrium. That's an electrochemical gradient. So in order for an ion or molecule to get across the cell membrane to diffuse, it can only happen by going through a membrane channel or going through the actual membrane itself through the lipid portion, okay? So either it can go straight through or it's got to take a channel, okay? So now that we've talked about diffusion, we need to talk about diffusion when it's water. So osmosis is the diffusion of water, okay? And basically in response to differences in the concentration of some solute, which is what's dissolving in a quote unquote solvent, which is water. All right. So osmosis is the diffusion of water. Osmosis is the diffusion of water. Osmosis is the diffusion of, all right. So characteristics of osmosis. Obviously, you're diffusing water molecules across the membrane. And it occurs across a membrane that is selectively permeable, like our cells. It's freely permeable to water, but not permeable to other solutes. Okay, so other solutes have a harder time getting through the membrane. So if the solutes can't get across, the water is going to go across. Okay, because that also will affect the concentration. So the water is going to flow toward that solution that has a higher concentration of solute. Because if you think about it, basically it's stuff and water. So if you've got a lot of stuff, it means you got relatively less water, right? Or a lower concentration of water. Or where you have not much stuff, that means you have a lot of water relative. So that lot of water is going to go toward not much water, okay? Now, This leads to things called osmotic pressure and hydrostatic pressure. So when you have osmotic pressure, it's the force of that movement of that water that's going into the solution as a result of your osmotic forces. Okay, so that's a lot of fancy terms for force related to osmosis. Okay, so force related to the fusion of water. Okay, you also have hydrostatic pressure, which is basically pressure in the fluid itself. And it can actually go against hydrostatic pressure, and it pushes against the fluid. Okay, so it's kind of like for every action, there's an equal and opposite reaction kind of thing. So osmotic pressure is pushing in one direction, hydrostatics pushing back. Okay, so not a basically everything is important. If that didn't make sense to you, you'll be okay. I'll just tell you that right now. So I do want you to be exposed to it, but I know sometimes those pressures can be a little hard to understand. All right, bulk flow. So when you buy things in bulk, what does that mean? You buy a lot of it, right? So water molecules are moving in large groups across the membrane. Okay, not so much one at a time. It's like a whole big group of water molecules go across the same time. That's bulk flow. Okay. All right. Osmotic concentration and osmolarity. Okay. So basically this just means the total concentration of the solute, the stuff that's in your aqueous solution, which is a fancy term for water, right? Because what is water in Spanish? Agua, aqueous, same kind of root word, right? So related to that, you've got essentially three types of osmotic solutions, essentially, or osmolarity. Iso means same, all right? So an isoosmotic solution is going to have solutes that are in the same concentration as the cytosol in your cell, okay? An hyperosmotic solution has a higher solute concentration. You like hyper, you think hyperactive, but really it's, when you think about hyperactive, means you're more active, right? So it's hyper is more. And hypo means less or low. So it's a lower solute concentration than the cytosol of a cell. And cytosol is the fluid inside the cell, okay? That's really all that is. So that sort of helps us understand tonicity, which is describing the effects of osmotic solutions on your living cells. Because osmotic, like that term could be for a living cell, or it could be just for anything. Right? It didn't have to be alive. Like if I have a beaker and I fill it up with salt water, there's going to be some osmolarity there because it's an aqueous solution, right? But if we're applying that to cells, we're now talking about tonicity. And so that same kind of root words we're dealing with again. All right, so we have isotonic, hypotonic, and hypertonic. So if there is no osmotic flow because the concentrations are equal, we call that isotonic. Okay. Now it says water does not move in or out. Technically, there's no net flow. Okay, it's just equal in both directions. All right, hypotonic means water is going to start flowing in the cell. Okay, and that can lead to what we call hemolysis. Right, because if water goes in the cell, what's that going to do to the cell? It expands, it swells. What happens if too much water gets in the cell? It explodes. Remember we said lysis? You might remember the root word for lysis or lytic? Okay, so anytime you see lice, typically we're breaking something. Okay, hemo is a word that means what? Anybody know? Blood, right? So like hemoglobin, right? So you're breaking a blood cell because it's got too much water in it. Okay. Now the opposite situation is when you have a hypertonic solution. And in this case, water is going to start flowing out. Okay, and that leads to what we call crenation. Okay, that means the cells start shrinking. Okay, they shrivel. Okay, so, and we'll explain this more in lab. So when we do this in lab in a couple of weeks, you'll get a much better understanding of kind of what that looks like. But you really mainly just need to know the terms and what they mean. So fluid. All right, so you can shift the fluid. So here you got osmotic movement in your water. It goes in or out of the cell. You get minor changes or fluctuations in your fluids inside and outside the cell, particularly in the concentrations of the solutes. All right, and those fluctuations get eliminated quickly. All right. Then you get system-wide changes in your solute concentrations that are going to take longer. Right, so if it's a small fluctuation, quick. System-wide change takes a while, right? Like if I want to change something in my classroom, that's my call. I can change it. If we want to change something here at the college, there's a lot of people that have to sign off on that, right? So it takes longer. Okay. So filtration. So filtration just means you have hydrostatic pressure forcing water across the membrane. Okay, so it's basically you're forcing the water to go across. It's not just naturally going across. And if you have large enough membrane pores or holes in the membrane, some solute can get across as well, right? Some molecules can come across. And in your bodies, filtration happens in some capillaries, or actually in your capillaries. It also happens in your nephrons and your kidneys, right? Because you want your kidneys to be able to filter so you can have urine, right? You want to be able to get out fluid and waste out. You want to be able to filter in your blood vessels so that, you know, when you get to the lungs, you can pick up oxygen, right? Get rid of carbon dioxide. You want to have filtration when you get to your capillaries get to your digestive system so they can pull that food out, right? To get it across to your body. Okay, so we talked about diffusion, we talked about filtration. All right, so let's talk about carrier mediated transport. So here you have integral proteins, and they're binding certain ions or substrates, okay, chemicals, and helping them get across the membrane. All right, so like enzymes, carrier mediated transport, these carrier proteins are specific, so they only work maybe with a specific substance that it binds and transports. They have a saturation limit, so once their spaces on their proteins get taken, they can't take any more until those substances get out. Okay. I mean, it's kind of like a bus, and your bus has so many seats. If you don't get on the bus, you gotta wait for the bus to come back after it's dropped off everybody. Okay. Regulation. So you have carrier protein activity that gets altered by other molecules. Okay. Like hormones. Okay. They can affect this transport through these carrier proteins. Now, carrier-mediated transport can move substrates, single molecule across the membrane, can move two substances in the same direction. We call that co-transport because they're going in the same direction. You can move different substances in opposite directions. We call that counter-transport, also known as antiport. So One substrate, one direction, two substrates, same direction, two substrates, opposite directions, okay? All right, so now if we want to break down carrier-mediated transport even a little bit more, you've got facilitated diffusion and active transport. Facilitated diffusion is just a fancy term that means diffusion with a facilitator, okay? So facilitator makes it easier, right? So compounds are going to bind to receptor site on that carrier protein. The carrier protein is going to change shape, and then the compounds get moved to the other side of the membrane and get released, okay? This works well for compounds that are not soluble in lipids. If they don't like lipids, all right, like glucose and amino acids. This is passive, so you don't need any ATP, okay? So the molecules are going to go, again, because it's passive, from high concentrated area to a low concentrated area. And once your carrier proteins get saturated, you've reached the limit of how fast your transport can go, okay? So once you hit that limit, that's it, okay? Because there's only so many spaces available. All right. So again, if you've got 100 people that you've got to get across, or get from the airport to the hotel, and your bus only has 10 seats, your ability to get them, it's really only affected by the fact that you've got 10 seats, and so you're going to have to make 10 trips. Okay, so once those 10 seats are taken, you're slowing down. That's as fast as you can go. All right, so basically we've talked about all the different kinds of passive transport that doesn't take ATP. Now we're talking about active transport. Okay, so you're moving substances across the membrane using ATP, and this is going typically against the concentration gradient, usually going the opposite direction. Okay, so in a sense, the concentration gradient doesn't matter. So examples of that are ion pumps. Okay, so these are carrier proteins, and they're transporting sodium ions, potassium ions, magnesium ions, calcium ions across the membrane. So most of them are going to move one of those cations in one direction. Okay, but some can move more than one cation at one time. And all these are cations because they're positively charged. Okay, remember that from chapter 2. All right, and you can also move one cation in one direction and one cation in the other direction. We call that exchange, right? Because you're one for the other. Okay, it's an exchange pump. Example of an exchange pump is sodium-potassium pump. Okay, you're exchanging sodium on the inside of the cell for potassium on the outside, and it requires ATP to do that. Again, I'm not going to ask you to be able to explain to me how the sodium-potassium pump works. I mean, if you want to take, a whole year of biology and a year of biochemistry, I mean, that's great, but I'm guessing, most of you probably don't want to do that, and I'm okay with that. So you just need to know sodium-potassium exchange pump is active transport, okay? Takes sodium that's on the inside of the cell, moves it out, takes potassium that's outside the cell and moves it in, okay? That's what you need to know. Now, there is also secondary active transport, okay? So here, the carrier protein is not using energy, but there's some other process related downstream that's going to need ATP, which makes it active. And it's usually to maintain equilibrium or homeostasis. All right, so you're going to move another substrate at the same time. Okay, concentration gradient in one substance is going to basically create the energy or the driving force for that protein to transport something else. All right, and so this could involve co-transport where they go in the same direction, could involve counter-transport where the molecules go in opposite directions. All right. So, all right. I think we're going to pause here. We can talk about vesicles next week. But what I would like for you to do real quick before we do exit tickets, I would like for you to talk to your partners about diffusion and active and passive transport. All right, so let's do that now.